Okay, so in this video I'm going to go over the answer to the course packet question from class 13. And the first part of the course pack question was asking about what happens if there are mutations in certain parts of the gene that are upstream of the transcription start. So the transcription start is here, and we're looking at regions either in the core promoter region, this is where the TATA box and things like that would be, or in this upstream region, which might be things like enhancers and silencers or cis elements in the DNA. So in this experiment, what they did was delete one region of the upstream region or of the promoter, and then look to see how transcription was affected. So in a normal cell and under normal conditions, the transcription rate would be 100%. And so then when they did a deletion, they would look to see if it was greater than or less than that 100%. So if we look at mutation A, which is located here in the upstream region, when this region is deleted, transcription is still 100%. So the conclusion that we might make about that region of the genome is that it is not involved in transcription of this beta globin gene. Then when we look at region B, so this is part of the promoter region, and when that part is deleted, there's less than 1% of normal transcription. So the conclusion you might make from that experiment is that region B is an essential region of the promoter, and that it's required for transcription of the beta globin gene. Region C, again, 100% transcription, so that's probably not involved. Region D actually has increased transcription, and what that would suggest is that region D could be a silencer region. So normally, when that region binds to a repressor protein, it decreases the amount of transcription. But when there's a mutation in that region, maybe the repressor protein cannot bind, and therefore there's excess transcription of the gene. So that's one way that it could have more transcription. And then region E, which is also in the upstream region, has about 45% of normal transcription. So that region could be an enhancer region. So normally an activator protein would bind to that to enhance transcription. When there's a mutation in that region, the protein maybe can't bind or doesn't bind as well, and so you don't get as much enhancement of the transcription, and you only have 45%. And then again, region F has 100% transcription, so it's probably not involved in transcription of this gene. So overall, the conclusions we might make from this are that which of these regions could possibly lead to this disease, and it could be region B, region D, or region E, because all of them affect the levels of transcription, and some increase it and some decrease it, but overall what's really important is that we have the correct amount. So for this disorder, having an unequal amounts of alpha and beta can lead to the disease, and any of these three mutations could alter that that amount of beta globin. So in the next part of the question, we're looking at a northern blot. So it's really important to remember that a northern blot allows you to visualize mRNA. So the bands that we're seeing are mRNA. And in this figure, these are where the lanes are located. So just to make it very clear, the lane labeled normal on the far right, so where there's this arrow pointing, this band is what the mRNA looks like for a normal person, or a person who doesn't have this disease. And then the numbers 1 through 5 represent patients that have this disease, and this is what their alpha and beta globin look like. So alpha globin is down here on this bottom blot, and all of the people, including the normal person, have bands for alpha globin. Some are just larger or smaller than others. So if we were to look at the first question, which is which type of thalassemia would you expect each of the patients to have? Patients 1, 2, and 3 all lack beta globin, so it's most likely that they have beta thalassemia, and that since they have no beta globin mRNA, that they're unable to produce beta globin protein. If you look down at the alpha globin blot, you can see that they also have slightly different amounts of alpha globin than the person without the disease. So it's possible that something is going on there too, but it's most likely going to be beta globin. Patient number 4 has a beta globin mRNA that is actually shorter than the normal beta globin mRNA. So here is where the normal beta globin mRNA runs, so it's right where this arrow is, and patient 4 has a shorter mRNA. You can see that they both of the bands for the normal individual and the person with the disorder are about the same thickness, so that suggests that they are both able to transcribe the gene normally. There's just something wrong in patient number 4 that causes that mRNA to be shorter. And then patient number 5 has a beta globin gene that is of normal length and of normal amount. So it's really hard to say, based on just this data, whether that person has alpha or beta thalassemia because they have normal amounts of RNA for both. And patient 4, it's most likely that this person has beta thalassemia because their beta globin gene is shorter. Okay, so now if we were to look at what type of mutation it might be that causes this disease in patients 1, 2, 3, and 3. So for them, they do not have any beta globin mRNA. So the type of mutation would be something that inhibits transcription or doesn't allow the gene to be transcribed at all. So these could be things like a mutation in the promoter, so a mutation in the promoter region, maybe a mutation in an enhancer. So it's something, or in another protein that would perhaps condense the DNA, so maybe something that causes DNA methylation, or something that affects the acetylation or deacetylation of the gene, so the HDACs and the HATs. So patient number 4 is the one that has the mRNA that's actually shorter than the normal length of the mRNA. So what kind of mutation could lead to that? It could be a mutation in a 3' splice site. So a 3' splice site mutation could cause an exon to be skipped that normally isn't. So that could lead to a shorter mRNA and also to the disease. There could also just be a really large deletion in the gene that would make the gene shorter. It cannot be a nonsense mutation. Remember, a nonsense mutation changes a normal codon to a stop codon, but that does not affect transcription, so that gene would still be the same length. It would just produce a protein that is shorter. Okay. So what type of mutation might lead to the result that we see for patient 5? So patient 5 has normal levels of alpha and beta globin and normal amounts, but they have the disorder. So if they had beta thalassemia, they could have a mutation that's within the coding region, and that could be something like a missense mutation, so that could change one amino acid to a different amino acid. That could affect the protein. It could be a nonsense mutation, so it could lead to a stop codon. We don't know what the protein looks like for this person, so it's really hard to say just by looking at this data. But if we wanted to know experimentally what type of mutation this person had, the easiest way to do it would probably be just to sequence the gene. And if we knew the sequence of the entire gene, we could then compare it to the sequence of a person with the normal gene, and then we could see if there was a missense mutation or a nonsense mutation. Okay, so now if we were looking at a mutation that would lead to mRNA expression but little or no protein product, this could be a place where perhaps there's a mutation in the start codon. So in that case, there would be no start to translation. There'd be no protein product. Another example might be, what if there was a mutation in a microarray so that caused it to bind to this mRNA when normally it doesn't. So remember, microRNAs are those short RNA fragments that bind to the 3' UTR of mRNAs, and they block translation. So if there was a mutation in a microRNA so that it could now bind to this mRNA, it could block translation of this gene. And finally, if we were looking for what type of mutation might lead to mRNA expression, but then a rapidly decreased amount of protein. So the protein's made, but then it's rapidly degraded. This could be some type of mutation within the protein, so that would be something like a missense mutation that maybe changes one amino acid to a different amino acid, and that change could cause the protein to no longer fold properly, and that misfolded protein could then be targeted for degradation by the proteasome. A nonsense mutation could also do that, so a truncated protein might also have problems folding and again be targeted for degradation by the proteasome.